Hello, I'm Clayton Mann, and my presentation is going to be on the effects of vitamin A supplementation on retinal dystrophies based on genetic variants. So to give kind of a quick overview here, I'm going to talk about the eye a little bit, narrow that down more specifically to the retina, talk a little bit about a couple of articles I used for my literature review, and then talk a little bit about some gaps in the research that I found that kind of drew my interest into the topic, and then talk a bit more specifically about my study. So, I think it's important when starting with the eye to think that the eye has all these different structures. It has, the sclera, the cornea, lens, retina, and all of these pieces are extremely important because they all come together to give us our effective vision. And so they're all part of something much bigger. But I think it's important to realize that all these smaller macro-sized structures have their own smaller pieces as well. And these structures are proteins and enzymes that help regulate and keep all of these macro areas functioning. So this is kind of one of the key aspects of my study because it looks at the quality of proteins and enzymes, which are of course dictated by genetics. So if you have damaged DNA in a section that codes for a particular protein that goes to any of these, then you could have somewhat failure in these macro-sized areas of the eye. And if those small areas fail, and then the bigger area fails, and then eventually your whole vision can just kind of have issues and have problems. So narrowing that down more specifically to the retina, which is the focus of my study, the retina is made up of this retinal pigment epithelium, which is shown by this sheet structure here. And what the pigment epithelium does is it absorbs photons from light. And photons are kind of like this light energy. And as soon as that comes in, it binds to this retinal binding protein, which retinol is technically vitamin A. They're kind of interchangeable. And after the binding of this RBP protein, it starts this cascade, which is actually known as the vision cycle. And this vision cycle plays a big part in us determining how much light is entering the eye and making conformational changes based on that. And so you can imagine that anywhere in this cycle, if you covered up, this protein or that protein, or stopped any of these transmissions, or somehow interfered with the cycle, then you wouldn't be able to complete the cycle effectively, and your overall vision could decline. And that's one of the important aspects of the study. And so whenever you're thinking about different retinal disorders, there's a lot of different things that you could choose from. And the reason there's so many different ways is because your genetics plays a part in what type of proteins are going to be affected and what the best way to treat those different areas are. Because you wouldn't give the same treatment for macular degeneration in all cases that you would for retinitis pigmentosa. Maybe some of them would, maybe some you wouldn't, but it's going to depend on those two different disorders at a base level. And so, looking at the literature review that I think highlights this point is, I found a study that did a knockout experimental study in mice where the RBP protein up here was actually removed and replaced with a generic sequence in mice. And the mice they ended up testing developed proteins similar to this that instead were the result of a frameshift type mutation. Now this is a limitation of the study because it doesn't tell us about what happens if there's a nonsense mutation or a missense mutation or anything like that, but it does tell us a little bit more about this protein's role in this cycle. And the people who did this research discovered high levels of retinol were stored in the liver whenever this enzyme was knocked out. And what this pointed to was that without this enzyme being present, retinol can't be sent to tissues and other areas where it needs to go. And as a result, vision was reduced in these knockout mice from this protein. So, highlighting kind of another area that focuses more on how treatment plays into the genetic role of this. In this study, they were looking at vascular endothelial growth factor and how polymorphisms affect its treatment. So, to kind of give you a little bit of terminology before we dig into this, polymorphisms are areas in the genetic code where there is normal variation from person to person. So you wouldn't consider someone with a polymorphism to be a mutant or have a mutation or abnormal, but you would consider them just to be them. It's kind of a bit of what makes them who they are. So in this study, they focused on vascular endothelial growth factor, and it's important because it promotes blood vessel growth in the eye. Now, you might think, oh, well, it's probably good to have blood vessels there, and it is. However, the problem is that excessive production can actually cause this condition known as macular degeneration. And the macula is part of the retina. And so, in this study, it was a retrospective study where they looked at old research on the topic, and they tried to determine if there's any polymorphisms that they could identify where it seemed that those improved the amount of treatment, or they improved what treatment should be done. And they found a couple results that they thought could be significant, but they didn't find themselves. However, they did find one very significant result, which was this RS833061 area. And they found out that this particular treatment, this monoclonal antibody treatment here, best fit for this type of polymorphism. And the reason why is because this A site that they found this polymorphism in was better targeted by this type of treatment than others. And so it kind of goes to show you that you might choose your treatment based on the genetics and based on how certain proteins will be affected based on a genetic basis, which I think is kind of the core of my study. And so the things that kind of led me into this was that there's lots of gaps in research. So as far as inheritance patterns, there's of course dominant and recessive, there's X-linked, there's all kinds of different inheritance patterns, but many people aren't really sure on what some of them are. For instance, retinitis pigmentosa is known to have dominant as well as recessive forms. There's a lot of questioning on whether some of these disorders don't concern just one gene, but are actually epigenetic and concern multiple genes. The other thing to consider is proteins involved. Even though many proteins in the vision cycle are identified, it's hard to say whether we have identified all of them. And, of course, there's many variants out there that are unaccounted for. Very different, changes in genes, such as the ones here, that people can't really say for sure if they're bad, good, or really what they are. And then probably another interesting thing that plays into it is that some variants cause a buildup of metabolites. So for instance, vitamin A, if it's not processed correctly due to some variant causing a protein that can't break it down effectively, these metabolites can actually build up, and even though vitamin A is routinely prescribed to patients with retinal disorders, because it plays a major role, it can end up being toxic to patients if they can't break it down effectively. So you might ask where I come in with this, and the whole idea of my study is that I believe that there's varying degrees of reaction to vitamin A. I believe that based on what variants you have, you could see fast improvement, mild improvement, moderate improvement, or maybe even no improvement or even worsening of symptoms based on what variants you have. And that's something I kind of want to bring more light to on some of these more unknown variants. And so the way that I mean to do this is by using a list of patients with various retinal dystrophies from Duke Medical Center. And these patients have already been identified to have genetic variants. Some of them are well known in the literature and others are not. And the plan is to record the variants and determine if they cause a pathogenic effect on vision. And using specific criteria, I'm going to look through the literature and compare these variants to similar mutations and try to identify if these really do cause problems. And the overall goal, of course, is to look for trends to see if there's particular variants that react a certain way to vitamin A. And so, to kind of give a bit more description of my study, it is going to be a controlled before and after study because subjects are going to be assessed before and after treatment occurs. There are going to be control groups because there will be individuals with variants that are known to be hindered or helped by vitamin A. And it's also going to be mixed methods because quantitative data is going to be used from ophthalmological examinations, but there will also be qualitative data from how patients describe their own vision. And here are my references, and thank you very much.